Hello, everyone. I'm Shahab. I'm a postdoc in Blake Richards' lab at McGill and Mila, and I'm so happy to be here. I'm going to talk about modeling specialized pathways of mammalian visual system with artificial neural networks. Okay, so I'm so happy that I'm talking after Tonio, because now I can basically skip all the introductory materials and I can just jump into the main things. But yes, I'm also working in this intersection of neuroscience and deep learning. And basically we use artificial neural networks as models of the visual system in the same way that Tonio described. So exactly as we saw before, this work by Dan Yammins and Jim DiCarlo has been one of the main and most successful works in this domain of so-called neuro-AI. So in this work, Yammins and DiCarlo showed that if you train a neural network on object categorization, that model actually turns out to be a very good model of the ventral pathway in primates, in the sense that you can use actually different layers of the model to predict the responses of different areas of the ventral pathway to different visual stimuli. But in this type of work, usually the visual system or the ventral pathway specifically is assumed to be a purely, strictly feed-forward neural network, one pathway, while the reality looks more like this. As all of us are probably familiar with this famous figure from Fleman and Van Essen work, the visual system has a modular organization. It has parallel pathways, and there are functional specializations for different modules in this network and for different pathways. Also, there is another property that I'm calling, I'm calling anatomical determinism, which is a very important concept for what I'm going to talk about in this talk. It has basically to do with the fact that across individuals, a specific anatomical location is always developing the same functional specialization. So I'll get to that in a bit. Okay, so with all the tools that we have from the Big Brain project and similar resources, with the data and tools that you all have been developing, now we can actually build models of the visual system that are more complex. We can use the anatomical data to set the connectivity between different modules in this sort of models, as Tonio mentioned in his work. We can also use psychotectonic data to, for example, set the cell types within each module and set the specific connectivity patterns across these cell types. So this all information can be incorporated in these models, and we can build in architectures informed by brain anatomy. We can train the neural network with one or a combination of ecologically relevant objective functions using gradient descent and error back propagation. And then we can compare the trained ANN with the brain. We can go back and forth between these steps and try to maximize the similarity of our model with the brain. And voila, you have a model of the visual system, and we're happy. We can probe it. We can do all sorts of experiments. But as I was working on this framework for the last couple of years, I realized that there are actually some challenges that one has to deal with while working in this framework. And today I'm going to focus on one of these challenges that has to do with that concept of anatomical determinism. So if you build this model of the visual system that is anatomically inspired by the brain, then we will have modules like, for example, V4, right? So this V4 module in our network should develop the same specialization as the V4 area in the brain. And every time that we train the model, we expect that the same sort of specialization happen in this area. And V4 as an example, in all the other modules, the same thing should happen. So this is not a trivial thing to do. Actually, with the current techniques that we have, depending on the initialization of the network, we actually get a lot of variability when we train the model. Sometimes MST might develop V4 like a specialization in our model. So we were trying to solve this problem in our work. But I will focus on a simpler version of this problem that has to do with the dual pathways of the visual system, the dorsal or the where pathway and the ventral or the what pathway. So we know that the ventral pathway is in charge of object categorization, scene processing, face recognition, and so on. And the dorsal pathway is in charge of localizing objects, tracking moving objects, reaching, grasping, guiding actions. And if we develop a neural network of the visual system, we actually want the neural network to have the same sort of specialized parallel pathways with the same sort of functional specializations. And ideally, we want to make sure that when we are calling a component of our model the dorsal pathway, we want it to develop dorsal-like a specialization. So this is the main challenge. In our previous work, we showed that if you take a neural network, an artificial neural network, and we train it with prediction, with visual prediction, the model receives a stream of natural videos, and the model is trained to predict what happens next in the video. So if you train a model like that, we will actually get representation to the what and where representations similar to the dorsal and ventral representations in the visual system. Specifically, if our architecture has two parallel pathways, each pathway learns different representations. One of them becomes a dorsal-like, the other one becomes a ventral-like. So we compared our model with mouse brain that's similar to primates and other mammalian brains, has this ventral dorsal pathways. And we observed that indeed one of the pathways shown in blue here becomes similar to the ventral pathway. So the plots here are similarity with brain areas. So the blue pathway becomes similar to the ventral areas of mouse visual system, and the red pathway becomes similar to the dorsal areas of mouse visual system. So in addition to comparing with brain, we also functionally probe the pathways of our model. Here we are using a motion direction discrimination task, which is a task that we know very well that the dorsal pathway in the visual system is specialized in. So there are a bunch of dots moving in different directions, and the model should basically report the direction of moving dots. Okay, so it's hard to see here because of the sampling rate of the videos, but it's basically either up, down, left, or right. And we observed that the pathway, the red pathway, the dorsal-like pathway, which is the pathway that was most similar to the dorsal areas of mouse visual system, does much better on motion discrimination compared to the other pathway, to the ventral pathway. So this is the task that throughout the talk I will use for probing which pathway is the dorsal pathway of our model. Okay, but as I said, in this model, the two pathways are identical. Okay, there's no difference between them. There's no interconnection between them, and only at the very bottom and the very top they are connected. Okay, so because these two pathways are connected, every time that you train the model, every time that you instantiate the model randomly, you actually, one of the pathway will become the dorsal pathway, the other one will become the ventral pathway. So, and this role will switch randomly from one initialization to another initialization. And so this is actually very unlike the brain. And so this is something that we wanted to fix. We wanted to say that, okay, in our model, the left pathway in that schematic is a dorsal pathway. And I want to show that this pathway always learn the same specialization. Now, this graph here is actually showing what I just said. It is showing that in every time that I initialize this network, one of the pathway will become the ventral pathway, the other one will become the dorsal pathway. So this is the difference in the performance of the pathways in motion discrimination, okay? And each circle here is actually one random initialization of the network. And you can see across like around 40 initializations, half of them, in half of them, path 1 is a dorsal-like. In another half, path 2 is a dorsal-like. So there is a randomness, and it's very unlike the brain. So we are looking for potential solutions, right? At the end, we need to create some sort of asymmetry between the two pathways, right? There are two sorts of solutions. One of them is basically creating some top-down asymmetry. I'll not go into that, but instead I will focus on initialization asymmetry. So we are initializing the two pathways randomly, but just by chance, there is an asymmetry between the two pathways. It's like noise, but still it exists, and it affects the function of the pathways. So at initialization, there is an asymmetry between the two pathways. One pathway prefers to become the dorsal-like pathway right at the beginning, okay? And we ask, what are those initialization asymmetries, and how can we intentionally create them, right? Because we want to make sure that the pathway that we want becomes a dorsal pathway. So we have to be able to create that asymmetry. So first, is one of the pathways more dorsal-like at initialization? So to test that, we initialize a two-pathway network, and we basically test one of the pathways on self-motion loss. So as you can see here at the bottom, the training data is some simulated videos in which a virtual agent is moving with some velocity and direction, and the model has to predict or estimate the self-motion parameters given the stream of input videos. So we test the performance of the pathway at initialization without any training. We do the same thing for the other pathway. And here I'm showing you the difference in, oh, sorry. Yeah, basically you saw the main results. So here I'm showing you the difference in the performance of the model of the two pathways on motion discrimination after training with prediction. And in the x-axis, I'm showing the difference in self-motion loss. Okay, at initialization. And you can see that there is a significant correlation between these two things, which means that at initialization, one of the pathways has the potential to become the dorsal-like pathway. And throughout predictive training, that potential is actualized. The other thing that we looked at is, okay, I'm running out of time. So the other thing that we looked at is the temporal convolutional kernels of our model. We specifically look at the so-called temporal integration constant, which basically tells us if a kernel is more of an integrator or a differentiator. Long story short, we also observe that there is a strong correlation between the temporal convolution properties of the two pathways and which one becomes a dorsal-like pathway. Okay, so with all these things, we can see that at initialization, there is a difference between the two pathways. But how can we create these asymmetries? So one thing that we know, based on the experiments that we have done, and I'm not showing here, handcrafting these properties is not effective. Okay, so we can't really go there and try to engineer asymmetries between them. And the idea is that the asymmetry is distributed throughout the two pathways. And usually, in order to create distributed asymmetries, handcrafting is very difficult. This is why backpropagation actually works so efficiently, because it's not about handcrafting. So our approach is to first pre-train one pathway on self-motion, only for one epoch, very briefly, which basically pushes one of the pathways slightly toward dorsal-like specializations. And then we take the two pathways and train both together on prediction. Okay? And so this is what we did. We take one of the pathways, pathway one, pre-train it for one epoch with self-motion. Then we take the two pathway, train both with prediction, and we observe that now across almost like the majority of our initializations, now the pathway that we pre-trained with self-motion now becomes a dorsal-like pathway. So now we have this tool to actually set which pathway to become the dorsal-like pathway. We do the same thing for the other pathway, and again we see that now across almost all the seeds, the random initializations, path 2, which was the initialized, the pre-trained pathway, now becomes a dorsal-like pathway. So with this, we have some sort of control on which pathway of our model or which module of our model will become the dorsal-like, or in general, we'll learn the specializations that we want them to learn, okay? So I'll summarize. Building in architectures based on brain anatomical data is the general goal. We need to anatomically match specializations between brains and ANNs. That is not trivial. Requires creating functional inductive asymmetries between different modules of our network. And we show here that initializations of the network matter. And in the last two remarks, I want to look at it from a broader point of view. we look at initialization as an algorithmic model of genetically induced asymmetries in brain circuits before eye opening. Okay, so this is something that we're working on, and hopefully we will have more stuff to present on these theories. I will take advantage of this situation to also tell you that I'm opening my lab at University of Montreal in January 2023. I'm looking for students and also collaborators to work together on these ideas, plus the using big brain data and other similar data sets to addressing these sort of questions. And with that, I thank all my collaborators and the funding agencies. Thank you so much.